Start the session. Kindly join the online candidates.
Yeah, go back to the previous slide. Let me see. Okay, good. Yeah. Hi, good afternoon. You all must be tired of seeing me last 2 days. Bear with me today and tomorrow morning. After that, I'm done. Okay, you're going to have new faces to face. Okay. My original idea was to give you guys a taste of what is synthetic biology. Okay. And then I came to know that not all of you are biologists. They have engineering background. So I wanted to go back to basic. So if some of you already know this basic thing, bear with me because you are familiar with it, but some people are not. Okay. So before we jump into synthetic biology, I wanted I'll give you some basic information is about the DNA and RNA and what makes DNA, what makes RNA, how the system works. Okay, so if you look at all forms of life, DNA carries the code for everything you do. Okay, that makes the core of your being, and RNA is a messenger. It takes the information from DNA, carry the code, take it to a place called ribosomes, and that's where the proteins are made to make your cell function. Okay, so let's talk about what makes the DNA, what makes the RNA, and we call DNA, it's called deoxyribonucleic acid. That's why it's called DNA, and RNA is called ribonucleic acid. And the thing that makes them is this thing we call nucleotide. The nucleotide has three parts to it. It has a nitrogenase base, the compound that has nitrogen in its structure, and it has sugar, is a pentose sugar, 5-carbon sugar. The DNA has a deoxy sugar, RNA has a ribo sugar. Okay, there's a slight difference in sugar structure. And then it has a phosphate molecule. All the three things together we call nucleotide. Okay, the nucleotide varies based on the N, the nitrogenase base. Okay, nitrogenase base goes like this. Wherever there is a letter A, the name of that base is adenine. Wherever there is a letter T, the name of the base is thiamine. Okay, wherever the letter is C is cytosine, and G is guanine. Okay, there are only 4 letters, right? The code is carried by this nucleotide. Adenine always pairs with guanine. Cytosine always pairs with, adenine always pairs with thiamine, cytosine always pairs with guanine. A, T, C, G, that's how they pair. They cannot cross pair. It's called complementary base pairing. Okay? So that's what you're seeing here. So look at the DNA. DNA is double stranded. It's like a ladder. Okay, you have a rung and a step. RNA is single stranded. Okay, so if you look at it, you have this the sugar, deoxyribo sugar phosphate that makes the ladder, side of the ladder. The step of the ladder is made by this nucleotide A, G, C, A, T, C, G, and so on. Okay, and the bond that connects one side of the ladder to another side of the ladder is the hydrogen bond that connects them. Okay, so this is how the DNA is put together, and it is at a double-stranded helical shape. It's all wound tightly together. Right? RNA is a single-stranded. The difference in RNA is RNA does not have the nucleotide thiamine. Instead, it has uracil. Okay, the letter U, and all other nucleotide is the same. So you replace wherever T in RNA, you replace with U. So instead of A, T in DNA, in RNA, what you will have? AU you will have. That's it. Right? That's simple. So that's the name of this adenine, guanine, cytosine, thymine. These molecules are classified as purine and pyrimidine. The purines are your adenine, guanine, pyrimidines are thymine, cytosine, and uracil. So that's the structure of the nucleotide. If you look at there, nitrogen in its structure, that's why it's called nitrogenase base. Okay. So you got that structure of adenine, guanine, there's a double ring structure. Pyrimidine are single ring structure. Okay, so the purine always pairs with pyrimidine. Purine and purine don't pair. Right? That's why it's called complementary base pairing. Okay. When you put all this together, that's the name of the nucleotide. DAMP, CAMP are the monophosphate nucleotide, the adenine, thymine, cytosine, guanine, and they all put together in this fashion. Okay, it's a helical shape, and you can see everything I just mentioned. Ladder and the step, and the hydrogen bond then connects them to make your DNA. It's a double helix DNA. The discovery of this by Watson and Crick, they won a Nobel Prize for that. They're designing how a DNA looked like. Okay. RNA, as I said, as uracil instead of thiamine. That UMP is the difference in nucleotide in terms of DNA and RNA. Basic structure of DNA, it's a double-stranded, a complementary base pairing, AT and CG. Antipolar, what it means is when there is one side of the ladder where it combines with the sugar and the phosphate, and we call that 5' end. That means the fifth carbon bonds with the phosphate, and then the other side is 3' end. So one side of the ladder starts with 5, has a 3 carbon where it bonds. The other side of the ladder is opposite, 3' and 5', 5', 3'. That's what that antipolar means. Okay. When it is helical, it just wound together, and there is the distance between each of those nucleotides is 20 angstrom, the small amount of distance, and then the perpendicular distance between these two ladder is 3.4 angstrom. So that's how much distance it is between the ladder, between the step. That's what it is. Okay. So that's how it looks like when you all put together. For class purpose, this is a cartoon of a DNA, but DNA actually looked like this, very, very condensed structure. When you take a skin cell and look into the microscope, look into your nucleus, it's all really, really condensed structure of your DNA. All the information is that one cell. Okay, you have all DNA in everywhere in your body, all the same. Okay, but the expression is different. They are expressed differently in different part of your body. In your digestive system, certain genes are turned on. In your skin cell, certain genes are turned on. But structurally, there is no difference where the cell comes from. That's what I explained, what is 5', 3' is. Okay. Now let's look at the function of the genes. So genes make up the chromosome of the cell, specific sequence of nucleotide, we call that genes. So when you have this DNA, they are very, 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 very long, all right? And then each small segment constitutes a particular gene. That particular gene code for particular function, okay? So if something happens, for example, you drank milk this morning, all right? And the milk has to be digested in your digestive system. So as soon as the milk hits your intestine, the cell in your intestine, in your stomach, and the intestine, what it does is the lactose in the milk is going to go bind to the gene that turn the enzyme lactase. Okay, the particular group of enzyme. That enzyme has to be produced in order for a milk to be digested. Okay, so that's how the genes are turned on and turned off whenever it is needed, whenever it is not needed. That's by design. So you don't want to have the gene expressed 24/7. That's waste of energy, right? So they have the a regulatory mechanism that turn your gene on, turn your gene off. Okay, so that is based on you have genes that are code for particular function that is called functional gene. And then we have structural gene, then you have control gene that control your switch, turning it on, turning it off. Okay, that's the sequence that turn the gene on, gene off, and then you have a gene that express for something. Those are called structural genes. RNA is single-stranded. As I said, U replaces T. The ribose sugar is different, and it's a tertiary structure of a protein molecule. Okay. So let's talk about how this control system works. As first discovered by these two guys, Jacob and Monod, both won the Nobel Prize for that. And they come up with this, how the gene turned on and turned up with a beautiful mechanism in E. coli bacteria. And E. coli, when you grow them in a flask and you give them lactose as a sugar, it produces the enzyme to digest the lactose only when lactose is present. When there is no lactose in the medium, the bacteria doesn't produce the enzyme. Okay, so they wanted to solve that problem, and they looked at all the gene sequences, and they come up with this set of genes that control, set of genes that express the production of the enzyme. So we have certain name for that, and that's how the genes look like here. So we have genes that code for expression of the enzyme is your the code that's a structural or functional gene there. Then this is your control part of the gene. So when there is a repressor that blocks your RNA polymerase, the gene is not turned on. So when you drink milk, the milk particle, one of the small part of the milk, lactose, going to go sit there, and it changes the structure of this repressor protein, it falls apart. So now when it falls apart, there is nothing to block for the RNA to express. That RNA polymerase will activate, and then your gene going to produce this messenger RNA, copying wherever there is T, you put a U, wherever there is A, you put a U, and so on, C and G, and complementary base pair, and it makes the expression of this small piece of messenger RNA. So it takes the code from this gene and makes this messenger RNA. This messenger RNA goes into a place called ribosome, and the ribosome reads the code, and that is called transcription. It is transcribing the code from DNA into RNA, what it means, okay? And then it translates the code by putting sequence of amino acids from two different amino acids that carries the sequences and put together the proper enzyme we call the protein, okay? And then when the protein is made and the lactose is digested, there's all the enzymes does the function of digesting. What is left is one small piece of lactose left, right? And the lactose will go back and that one lactose that is out there attached, right, is still there. All the lactose is digested, then one lactose is still there, and lactose also will be digested. Now this repressor protein goes back to its original structure. It will go and block that part again. So the gene is turned off. Okay? The molecule that blocks RNA polymerase, the structure, because of it tightly placed on the gene, the RNA polymerase cannot go and read, so it doesn't make messenger RNA. In order for that to be turned on, what do you need? The substrate. The substrate is a milk in this case, right? Lactose. And that lactose goes there, so it's changed the structure of your repressor protein, falls apart. It makes RNA, there's one lactose left on the repressor protein. Enzyme digests the lactose. The repressor protein goes back to its original structure and goes back and clamp on that region, that lock position. The gene is turned off. Does it make sense? Right? So you need to have an inducer to induce the production of enzyme. That's called induction. Okay? That's how the gene is turned on, gene is turned off. Okay? There's another mechanism. This is we call that lac operon because of lactose. Okay, that's why they express first publication to express how the genes are turned on or off. There is also another gene mechanism is everything is like in the word form. It's there. I already explained it. Okay. Then we another expression called arch operon. In this case, the opposite happened. The gene is always on. Okay. And when you overproduce the enzyme, it's called reverse happens. Okay. You change the reaction over again and stop the gene to go back to off position. That's called arch operon. We call that co-repression. The product itself, it turn the gene off. Okay, the enzyme that you overproduce will turn the gene off. That's called co-repression to shut. There are two mechanisms. One is gene is always in off position, you turn it on. One is gene is always on position, you turn it off when you note it. Okay, that's how, in simple term, how gene is turned on and turned off based on what is inducing the gene. We call that a transcription factor. The transcription factor is, in this case, in talking about milk, is lactose is milk. Okay, so some people have milk, lactose intolerant, right? They cannot digest milk. They have problem controlling this. That's why they cannot, produce the enzyme. So this is the opposite way called arch operon, how your arginine is produced and turned off, because in this position gene is always on, and then when too much enzyme is produced, and now it's going to go sit on this and go back to lock position. So that's just the opposite of control mechanism. So that's overall, all genes are controlled by operons, operator region of the gene we call operons, and there is various positive control and negative control mechanisms. Okay. All right. The problem comes when you have mutations, right? When you change the code, when there is in one place A is replaced with something, and it becomes reading that particular code become nonsense. It doesn't make any sense because codes are supposed to be read every 3 nucleotide sequence. That each 3 nucleotide makes a codon, it codes. But if for mutation, when one of the code changes, it misreads everything. That's where the problem comes. That is called mutation, a permanent change in the gene. Okay, that's a mutation. So there are a lot of different mutations there. I don't want to go bore you with that. All you need to know is the nucleotide sequence change and cause problem. That's mutation. Okay. So if you go through this spontaneous mutation, it happens spontaneously in your body, and then you have environmental exposure like radiation, UV light, all the gamma radiation you get exposed. Chemical exposure will induce mutation. Okay. These are we call that mutagens that cause mutation. We have physical mutation, chemical mutation, all kinds of mutations that change DNA structure. And then we have the different kind of mutation that the code switches because of deletion. For some reason, during the replication, DNA replication process, one of the nucleotide doesn't replicate. You delete it. That is called deletion mutation. And then insertion mutation. Sometimes a wrong nucleotide is inserted when the DNA is reproduced. That's called insertion mutation. Okay? So there's a lot of a name for it. It's in the PowerPoint, it's all the mutation is happening expressed here. This is the deleted piece of the DNA when they reproduce. This part is missing. So the code for something is not there. And that is deletion mutation. Insertion mutation is coming from some other gene, it's inserted into this gene. So that's called insertion mutation. Just explain how when you put a new code in the original place, there is a normal code over there, and then this code changes. See the cytosine, thymine, adenine, guanine. Now it's switched, right? So you don't have a C and A. In this case, this code is changed to instead of T and A, you got to have a C and A. Okay, that is a mutation. All it takes is one letter change, and everything going to change. Expression of your gene going to change. Okay. So it's going to produce a wrong amino acid and wrong protein in your body. You cannot digest, you cannot, protect yourself in some cases. Okay. Those are called frame shift mutation, the way so far I described, because you are reading the DNA in certain frame. The reading frame shifted. That's why it's called frame shift mutation. And then we also have non-frame shift mutation. In this case, whole chunk of DNA is, switched. And that's called non-frame shift mutation. That happens because of substitution mutation, inversion mutation, a lot of things happening, the whole chunk of gene is changed. Okay? There's another thing in your body, we call that transposable genetic element, and these are genes that doesn't have a permanent place in the chromosome. They are all over the chromosome, and they move from one chromosome to another chromosome, and that's why these commonly known as jumping genes. Okay, the genes jump from one chromosome to another. They don't have a permanent place on the chromosome. Okay, so that is called transposable genetic element. Okay, that's explained the non-frame shift mutation. So how the whole chunk of DNA is moved, a lot of letters change, and then doesn't make sense. Okay. There's so many of them, so we don't bore with you. The insertion sequence change, then transposon, we call that jumping genes. Jumping genes move from plasmid to plasmid in bacteria, plasmid to chromosome, chromosome to plasmid. They move all over the place. Okay. This was first discovered by agriculture scientist in Iowa State. Her name is Barbara McClintock. What we looked at the corn kernel is corn kernel, instead of 100% yellow, some of them are red, or brown, or different color. She was very curious. She worked to entire her adult career on that one, answering one question: why this corn has different color, right? And she said that's because of jumping gene. Okay, she got a Nobel Prize for that one work. She got the Nobel Prize. She never gave up. 40 years of her work. Okay, so she coined the jumping gene because she found out there is particular gene sequence. There is no permanent place for them. They are always moving, and whenever they move and sit on a new place, it's going to change the code. As a result, instead of producing yellow color corn, you're going to produce a red color and brown color corn. That was the reason that corn was different color, and that was Barbara McClintock's work. Okay, so that's her. Barbara McClintock in 1950 got a Nobel Prize in 1983 at the age of 81. Okay, her entire life she worked on the project. Okay. All right. So that's how a transposon look like. These are the pieces, don't have a permanent place. They move all over the place. Whenever they move and sit there, change the code and then cause mutation. Okay. And then recombination. What is recombination? Recombination is if you have gene from more than two sources. You have your own gene, and you had a gene from somebody else, put it in your body. That's a recombinant gene. Okay, so the recombinant gene happens in bacteria by these three phenomena. This morning I quickly told you: transformation, conjugation, transduction. It happens in us also by when you go for modern medical treatment, the gene therapy, right? The gene therapy is putting correct gene in your body. They take a gene from normal person and then put that in your. If you have a disease or something, genetic disease, they can correct it. And that is a recombinant gene, because now you have a gene, not only yours, you got a gene from someone else that has a correct gene, right? So that is called recombinant, recombination. The gene is recombinant gene, okay, recombinant cells. So in bacteria that happens naturally by these three phenomena I explained. I'm not going to go in detail. Transformation is from gene that free floating, the bacteria takes that's called transformation. That's how it happens. That is called transformation mechanism. So here is the gene freely floating, and this bacteria took the gene and inserted into the sequence now. Right? That is, now we got a new trait, new character for that. Conjugation is between two adult bacteria. They connect through a conjugation bridge, and they exchange genes. And so they connect, and a piece of a gene is donated, and a piece of a gene is inserted into the genome, and now it has new features. Okay? That is called conjugation. This is a real picture of a conjugation. Here is a bridge formed in bacteria in electron microscope, and this bacteria is changing the gene. It is taking the gene from this bacteria and putting it in this bacteria. This is called F plus male bacteria, F minus female bacteria is called conjugation. This bridge is conjugation bridge, F plus, F minus. They form, take a piece of DNA transferred here. Okay. Now this is a recombinant cell, because this cell has its own DNA and piece of DNA from this bacteria. Okay. So if it has two or more from different source, it's a recombinant cell. As the term F is fertility factors, and then transduction is viruses. When virus infect you, they accidentally will take a piece of your gene and transfer it to another gene in bacterial system. That is called transduction. It's an accidental gene transfer through a virus. That's called transduction. So that's how it shows. When virus infect a bacteria, it's accidentally take a piece of a bacterial gene and transfer into when the other reproduced virus infect another virus, it's going to inject bacterial gene, and this bacteria got a new piece of DNA from this bacteria. Okay. That is called transduction. So now when this bacteria get that gene from another one, this become a recombinant cell. Okay. So with this background, now we can manipulate the gene. We can take the genes that you really want for your useful product or cure for disease, insert into your cell. That is called genetic engineering. We are engineering the DNA, we are manipulating the DNA. There are some special enzymes out there that does this for you, and some of the enzymes called restriction endonuclease. This is simply, the analogy is scissor. It cuts the gene in certain places. Okay? And then we have a tape that connects the gene back. Okay? Those enzymes are called ligases. Okay? So we have restriction endonuclease will cut the gene, and ligases will put them together like a scotch tape. Okay? So that's what's happening. If you want to take the gene out, you take restriction endonucleases, put it in the bacterial system or human system, it will cut the gene in certain places. If there is a library of these enzymes, it will cut in specific places. You use that specific enzyme wherever you want to cut. It doesn't cut willy-nilly all over. Okay, it has a different system, so you need to use the right enzyme to cut it in the right places. Okay. Then DNA ligase's function is to put the gene back together. Okay. So that's what this picture is showing here. So you take a donor, you treat them with the restriction endonuclease is going to cut a piece of gene, and then you have the gene inserted into a vector. The vector is going to carry into a donor, in this case sometimes viruses, sometimes bacterial plasmid, and then you insert that donor into this vector, and then you put the vector into the bacteria, in the recipient cell with the ligase enzyme, and it will put it together, and you got the donor receive the recipient. That's using these two enzymes. You cut and join, and you got the new gene expressed in your recipient. Okay, so this is how nowadays insulin is made, right? Insulin is made in large quantity. Olden days you need to use animal model to make insulin. Now you use E. coli bacteria. So you put the human gene, insulin producing gene in E. coli, and E. coli will make insulin in big bioreactor, and you purify it, and that's how insulin is made. It's genetic engineering. Okay. And then another set of genes called enzyme called reverse transcriptase. Function of this is to reverse. You can make DNA from RNA. All you need is reverse the code, right? So wherever there is A, U in your RNA, you make a A, okay, to make a DNA, right? So that part is carried out by this enzyme called reverse transcriptase enzyme, okay? So when you use this enzyme, you can make DNA from RNA. We call that cDNA, complementary DNA, okay? And it complements with the code of the RNA. So you can manipulate with all these enzymes, you can manipulate these genes whichever way you want to make the particular product, okay? So to do that, you do this electrophoresis to see whether your gene is expressed in correct places. That's this instrument called gel electrophoresis. You make that gene alteration and you have your control, and then if your gene is taken by the donor cell, it has to match, the band has to match the control. That means your gene is in right place. If it doesn't match, because something went wrong in your experiment. Okay, so that is basically what this gel electrophoresis does. Then polymerase chain reaction. Most of you heard about this PCR. You kind of replicating that piece of a DNA in a test tube, basically, right? So that is called PCR reaction, polymerase chain reaction. So what you do is, because of this enzyme we talked about, you put them in the enzyme in the test tube, and first you get the piece of gene and you heat it up. When you heat it up, the double helix will fall apart, and then you have the primer of the gene, and then you cool, and then it replicates, it produces new piece of DNA, and then you cool, and then it forms the DNA again. You heat and cool, add more primer, you form several million piece of DNA in a test tube in a short period of time. Okay. That is called polymerase chain reaction. So that's how it works. You go through a cycle of take a piece of DNA and go through, you denature them by heating them, the DNA, the helical shape fall apart. Then use a small piece of primer that attach to it, and you have the DNA polymerase enzyme. The function of the enzyme is to reproduce the whole sequence back there. Now you got a 2 sequence code, and you heat again, and then it fall apart, and you add more DNA polymerase and make another set of DNA, and then you heat and cool several times, and you made millions of pieces of DNA in a small test tube. That is your polymerase chain reaction. So that's how it's cycle 2. It keeps going on. From one small piece of DNA, you make multiple copies of same DNA over and over and over again. Okay, heating and cooling, adding enzyme and primer. Okay, that's just to show you small piece of fragment. You doing that, you made it 64 copies in a short period of time in a test tube. Okay. We call that PCR thermal cycler. Everything is now easy to do. A high school student can do that. This, when it was first discovered by Dr. Mullins, he got Nobel Prize for this discovery, how to make PCR. Okay. Now with the thermal cycler machine, you take a piece and put in the test tube, insert it, everything is programmed. You push a button, 1 hour later, you got millions of copies of DNA. Okay, so that's called thermal cycler. Okay? So that's how the PCR reaction works. Then gene probe hybridization. If you wanted to pair with the known piece of primer, if you have a small piece of primer, you can hybrid. If you have a whole sequence, it will hybrid with them, because A match with T, right? C match with G. So if you have a small piece of DNA in a crime scene, and if you want to make sure that somebody has the same gene that committed the crime, you make the PCR reaction, put that in the test tube, and see whether it's going to match them. Right? If it matches them, that's a culprit who did the crime. Okay? So So that's how this works. That's called DNA hybridization using probes. Okay. So you do that and hybrid them. If the primer match, it's going to stick to them like a Velcro. It's going to go wherever there's a code, it's going to stick to them. We can also do dot plot by see whether it matches, it doesn't match. Gene sequencing, now we are done. We know human genome project, we all have 3 billion nucleotides that make human, and 20,000 genes that make a human gene. So those are the thing already done. So 3 billion base pair we have 20,000 human genes. So we can make a lot of products by knowing this basic information. So here's a piece of how you can manipulate the gene. So we can, in the overview of recombinant DNA technology, you can isolate the gene and then you insert it by using these two enzymes we talked about, ligases and the gene that cut the piece. And then you put together, now you got the recombinant gene, and then you put it in the bacteria. And every time when bacteria multiplies, the gene is multiplied. Okay? And then you got a new product. You can insert that into a plant, animal, wherever you want. So you got the genetic engineering tool now available for us. So there's more steps involved, how everything is monitored and altered. Okay? So we can make a lot of products. So this is a list of products already in market by genetic engineering, some hormones, some immune treatment, enzymes, vaccines, a lot of things now produced in commercially. Then we can do that in bacteria, we can do that in animals, we can do that in plant, we can make transgenic organism, any organism you can manipulate, you can make transgenic organisms. You put a foreign gene in them. Okay. So we can alter the gene and to the need, like degrading chemicals, producing new chemicals. So if you know the code for particular enzyme that you need to make a particular product, you can tailor-make all those things together. Okay? So transgenic plants, there are a lot of transgenic plants out there. So one example here is in this case they took a gene from an Arctic flounder fish because this fish can survive very, very, very cold winter in Arctic, right? And they put that in plant like strawberries. So when there is cold freeze, the strawberry plant won't get died because this plant can survive cold winter because of their Arctic flounder gene produces protein that protects them, okay, cold tolerant protein. Okay. So that's how we can make transgenic plant, finding right gene to put in right places. This is the schematic of how it is made, transgenic plant. Some examples of transgenic plant that we commonly use in tobaccos and other high-end crop. In here, an example of herbicide-resistant plant. Okay, so you put herbicide-resistant in the gene. When you apply herbicide, only weeds get killed. Your crop survives. Okay, and here's a pest protection. In this case, you took a bacterial gene and put it in the plant. Okay? So one common one is called, I don't know how many of you heard the term BT, Bacillus thuringiensis. Bacillus thuringiensis is the name of the bacteria. It's a natural pesticide. This bacteria produces protein that kills the pest. Okay? So what they did, they took the gene from this bacteria and put it in all the crops. Right? You have BT cotton, BT brinjal, BT everything now, BT corn. So all the product that I don't know whether it's a regulation here in the US, there is no regulation. Everything is genetically modified. Okay, so that's one example of pest protection. You took the small piece of gene in one bacteria, put it in a plant, and plant express the same protein. So you don't apply pesticide. Plant expresses the same protein. When pest bites the plant, the pest gets killed. Transgenic animals, so we make foreign gene, put them in the animals. and you produce a lot of product in this case. Okay. There are various methods to inject the gene, like electroporation, microinjection, gene gun, a lot of things you can use to inject. So a lot of product you make with the animal models. So we produce treatment for hemophilic patient, emphysema, certain cancer, growth hormone deficiency, heart attack, stroke, everything that need. These are the protein that you need, and you can express them in animal model, and you can use them. Okay? Gene therapy, right? Gene therapy is basically correcting a defective gene in a person, and get the donor, and then you put them in your bone marrow, and then hopefully your body takes the gene, and it's going to multiply in your body. It corrects itself, whatever mutation you have. That is gene therapy in short. So in order to do that, you need a carrier. The carrier of the gene is virus. They put that donor gene into a virus, mostly retrovirus. Retrovirus is what injected into your bone marrow. Okay. That's how it looks like. So you take normal gene from a normal person, isolate the gene, you clone the gene, you insert that into a virus, and then you inject this load of viruses into the bone marrow of the patient. And the bone marrow patient shall infect the retrovirus. The virus will transfer the gene into the bone marrow. Okay? And then transfected cells are reinfused into patient. Patient observe over time the gene is multiplied inside, and it's going to express the protein that need a normal protein, and you get cured. Okay? It's very in a condensed way to say how human gene therapy works. Okay, there are a lot of different other methods. It's just to show you how it works. Okay, so this is just a basic fundamental biology of how genes are manipulated, how nowadays your insulin is made, how corrected gene is put in your defective patients. Okay. So with that introduction, now we're going to move into synthetic biology part. Okay. Open that. So I added this part this morning because I thought you guys need a little bit of basic fundamentals. Okay. Synthetic biology, yes, yeah. So what is synthetic biology? Now synthetic biology is you can make the nucleotide yourself. You don't need any cells. You can make your own A, T, C, G. And chemically you can synthesize them. And you can design your product. If you know what gene sequence make insulin, what gene sequence makes certain enzyme, certain hormone, you can make your own gene tailor-made in a test tube. You don't need to go find a donor, you don't need to find somebody else. And that is synthetic biology. You're synthesizing your own code and put it in there, right? That's tailor-made, okay? That is, in simpler term, what is synthetic biology. You're synthesizing your nucleotide in basic chemical reaction. So synthetic biology broadly refers to the use of computer-assisted biological engineering to design and construct new synthetic biological parts, devices, and systems that do not exist in nature, and you redesign of the biological organisms, existing biological organisms. You can redesign the organism any which way you want. You can manipulate the bacterial cell to produce particular product. You can increase the yield. Anything you want, you can do. There is technology out there that is called synthetic. It is scary, right? It could take into a wrong turn, and you can produce a Frankenstein monster there. That is regulatory issue. That is not scientific issue. Academically, we can make anything, but regulators have to worry about that. Okay? So synthetic biology incorporates the technique I just told you, the molecular biology techniques. It differs from recombinant DNA technique. In recombinant DNA, you have to find the gene from somewhere, insert it, and put it in the donor. In synthetic biology, you don't need that. you know the code, everything is already known. For all the 20,000 genes that make a human, we know exactly where those genes are located, what are the function of each gene is, right? So if a person has some genetic disease, you can correct them. Okay. So it differs from recombinant DNA technology in that synthetic biology introduced synthetically constructed part, and it's not limited to the modification of the natural organisms. So you can do anything you want. Okay. Construction of life form with no natural counterpart. Right. People have created new viruses, new cells in the lab, right? So it never existed in nature before. You created new organisms. You create your own organisms. So it's parallel to engineering, right? In engineering, system biology aims to study natural biological system as a whole, often with biomedical simulation modeling tools in comparison with experimental formation. So relationship between system biology and synthetic biology is you observe what is happening in nature, how gene work, and study any problems it has, everything, and you correct it and make a tailor-made thing. Yeah. You know the natural system first, and then you make your own synthetic biology. The engineering design is design cycle and rational design in synthetic biology is parallel. Okay, yeah. Key aspect of synthetic biology is which differentiate from genetic engineering and current biotechnology approaches the application to biology of techniques which are normally used in engineering design. So we are taking engineering tool, combining with biology, and that's what synthetic biology is. Yeah, we use a lot of computer, a lot of computer modeling, and find out bioinformatics, and use those knowledge and create new systems. Okay. So let's compare how this works in engineering cycle. In engineering cycle, you first get your specification, right? What do you want to make? You have your specific, and then you design it. And then you model it, and then you implement it, and you make your product, right? Exactly same thing in synthetic biology, right? You are duplicating the engineering model in synthetic biology. So you have your specification, you have your design, you have your model, you produce your prototype, and then you produce a real product, okay? So let's differing approaches to synthetic biology. You have DNA synthesis at the most basic level. Synthetic biology involves the use of synthetic DNA that was uploaded, returned in a computer code, and printed out from a bottle of nucleic acid. This nucleic acid, we created them synthetically, not from nature. These nucleic acid codes are these 4 codes we talked about: adenine, thymine, cytosine, guanine, the ATCG, and you put them in a bottle in a designer way. You design them yourself. Okay, so these DNA strands are then inserted into organisms through a variety of genetic engineering techniques to create whatever the need that you need for. We also have biobricks. Biobricks are standard DNA sequence that code for certain functions. Those are called biobricks. Okay? So DNA sequence can be created to make an organism glow, light up, for example, and engineering that biobrick into an organism that should make it glow. Okay? So it's a piece of structure of the DNA that carries out the glowing function. You can do that. These open source bricks can be used by researchers across the world to construct new genes and DNA sequence. Okay, so most of these are open code available to the researchers. Minimal cell genome. So let's look at. I said people created new organisms. So the guy Craig Venter is an entrepreneur, as a scientist. Working to produce organisms or cells that with a minimum number of genes created a new organisms. Okay, one could then add any DNA sequence to this minimal gene or cell and produce anything you want, like fuel, you make medicine, or any other synthetic product. Okay, so this organism never existed in nature, right? And Craig Venter, the first guy to create that in the lab. Okay. So that's how it works. You get your in silico design of genome, put the nucleotide in the test tube, you assembly the nucleotide and insert a certain piece of that into existing organism, or you can also create a new organism. In this case, you are putting in the existing bacteria a right piece of gene to express right product. You insert that this nucleotide that never been in any organism, is you created in a test tube, and you insert that into this bacteria. And now the bacteria took that green color is the one that you inserted into the red color. Now it's become a recombinant. Whatever it codes for, now the bacteria are going to express them. Okay, that's one way of doing it with synthetic biology. Okay. We have xenobiology. Xenobiologists are attempting to create alternative genetic systems such as novel nucleic acid like suicide gene that mirror biology. So suicide gene is not to commit suicide, it to kill unwanted organisms. Okay, so that's called suicide genes. And so xenobiologists can create that. Okay, so it's like a mirror image of creating a nucleotide. It almost look like a nucleotide, but it's not the real nucleotide. That's what it is, the mirror image of that. Okay. So that's example of that. So for example, thiamine look like this, and 5-chlorouracil look like that. So you can substitute wherever there is thiamine with this nucleotide, that's a mirror image of that. It almost look the same, right? So that is basically what it is, this phenomenon we call. Okay? A team has replaced thiamine with 5-chlorouracil in the genome of E. coli, where others are attempting to create mirror cells. The cell exactly look like E. coli, but it's not a E. coli. It doesn't have a uracil. Then we have people created protocells. So evolutionarily, how the first cell produced in Earth, it's called protocells. People created in the lab now. Okay, so in the primitive atmosphere, early time when in the evolution period, these molecules are developed over a long period of time because of the natural phenomenon of lightning and organic chemical mixed together to create organic molecule. Inorganic molecule and lightning and various chemical reaction. So that's how we produce the first organic molecules, and amino acid nucleotides were produced, and later on it developed into RNA, DNA, protein. The people are now doing that in the lab. We can create the same thing how it all started in the lab. We call that protocell. So protocells are almost like a true cell, but they cannot reproduce. Okay? You have to synthesize them. Fundamental techniques in synthetic biology. Some of the fundamental techniques you need to know: computational biology, computational modeling, DNA sequencing, DNA synthesis. Okay? There are some technological enablers that have facilitated the emergence of this new field. So you can have the sequencing machine that can sequence any piece of sequence you want. These 4 letters is all you need. You can put in different combination for different product, and then you can put it in the computer. You get a finished sequence, okay? And the computer can do various permutation combination, and it will spill out all the different product with the sequence of different sequence code. Okay? So you have a computational modeling, DNA sequencing, DNA synthesis can facilitate synthetic biology. Computational modeling is synthetic biology approach. In this case, is the design of an engineered biological system through engineering cycle. The modeling of the design is to predict the performance prior to fabrication is an important component. So you're going to model, you want to know what it's going to do. You need to know what is the function of that. Okay. Synthetic biology is therefore similar to system biology in that both rely heavily on computer modeling of biological processes. Okay. So you need to know the code, right function of that gene expression, and make the right product. Okay. DNA sequencing, the reading of sequencing DNA is the second step in synthetic biology. Again, I told you the DNA comprises 4 bases. The entire content of DNA for a particular organism is called the genome. If you know all the code, the human genome, so that is all the genome, all the code that make a human, we call human genome. Okay, we have most of the agriculture important plant genome, animal genome, lot of bacterial genome are all now known, are publicly available. Okay, you can Google them. You can get in your in your phone. Who is that connecting back? Somebody is not authenticated. Okay. Okay. All right. So once the genome is sequenced, the next step is to rewrite and synthesize all parts of the genome. There are a number of cases where the genome of organism is entirely synthesized in the lab. Okay. There's no other model out there. They created one. Can you switch that? I cannot move. So, in short, by knowing all the DNA parts, I just explained these enzymes, DNA polymerase for replication, DNA ligase for putting the pieces that are broken, RNA primer, DNA primer, all these things that basic biology taught us, we can now manipulate and create new organisms. So this is your first viruses created in a lab, never existed in nature. Okay. So in 2002, this scientist Cello and co-worker in State University in New York synthesized a polio virus. Polio is eradicated almost. Because polio virus genome sequence is out there, you can Google, download what is polio virus made of and the sequence of polio virus. So now, all you need is the nucleotide sequence. Put them together, you can create your own virus. This group of scientists did that. Produced the first synthetic virus by putting the right nucleotide together, creating it in a test tube. In 2003, the particular virus, bacteriophage, was assembled just 2 weeks' time by a team of small group of people in Craig Venter. I told you the Dr. Craig Venter Institute created another viruses in a lab by putting the sequences together. Okay, so it's scary, and that is synthetic biology for you. So we can create anything you want if you know the code, if you know the right sequence. All right, let's look at application of synthetic biology. How we're going to apply. The ultimate goal of synthetic biology is to develop commercial application that benefits society. Okay, for example, to design and build, engineer biological system that process information and produce new products that are useful to us. Okay, that is the beneficial part of synthetic biology. So we can manipulate chemicals. For example, the chemical Farnesene, which is an essential building block for a wide range of chemical products like detergent, cosmetics, perfume, industrial lubricants, transportation fuels. That's farnesene is a building block of the chemical. So you can make that through synthetically engineered yeast. You create yeast with the right code. That yeast will make that chemical for you. Okay, that's a beneficial part of synthetic biology. You're doing the right useful product to make the society better. Okay, and you can maintain and enhance human health and also our environment. So here is the product, synthetic biology product. In California, they engineered yeast to produce this artemisinic acid. This fights as an anti-malarial drug. Okay, the research and companies are now working to produce vaccine for malarial vaccine using synthetic biology. So this is a different story. The artemisinin was accidentally discovered as a malarial treatment process. Artemisinin, these guys are trying to do synthetic biology to make jet fuel. Okay, but put a wrong code and they ended up making different product. Okay, has become a useful product. Okay, and then they went back and corrected. They made jet fuel. And this is the byproduct of making a mistake. Then you can re-engineer like type 3 secretion system from Salmonella bacteria to make your lightweight spider silk protein, to make lightweight bulletproof vest. The cops wear bulletproof vests that are very, very heavy. Nowadays, bulletproof vest is very light. In the US, everything is made up with this spider web protein. The protein comes from spider. The spider that webs make the web, that's a protein. The same protein, the gene is put in the goat, and the goat produces the milk. The milk has that protein. You get that protein out, and you make that spider web, make that bulletproof vest. How about energy? Organisms synthetically engineered to break down biomass into sugars for fuel. Algae naturally produce oil, but through synthetic biology tool, algae can be re-engineered to produce oil that are chemically similar or identical to the oil that are currently used in today's transportation and energy infrastructure. So that is possible in synthetic biology. You can create organisms that make the product that are very, very similar to natural product. The organism can create the product for you. So in synthetic biology, that is possible. Natural product substitutes you can make. Synthetic biologists are working to replace some of those substitutes. Synthetic production of rubber through isoprene, a crucial building block for making artificial rubber. The gene code encoding the isoprene, previously found in rubber tree, right, has been synthetically engineered and put in an E. coli. Now he can make this rubber by making this bacteria express that protein that you can make the isoprene, the building block for your rubber. Okay. Other products currently being produced through synthetic biology include vanilla, stevia, and palm oil, among other things. So stevia is your pure essential oil, and these are products already made using synthetic biology tools. Okay. Impacts of synthetic biology on the conservation and sustainable use of biodiversity. When you release a synthetic microbe, synthetic organisms threaten biological diversity. If they escape into the environment, either intentionally or unintentionally, we don't know the consequences, what that could happen. That's one fear about this. It's release and mingle with the natural, genome in the wild. You introduce the synthetic genome, it can create something totally that you don't want, that can end up making that. this very quickly, the COVID story, basically synthetic biology of the mRNA vaccine is basically the product of synthetic biology. Okay, so COVID-19, I don't want to go back and relive all that thing. So this is how we want to make a vaccine. It takes a long time to make a regular vaccine. Testing and all that takes really, really 20 years to make a vaccine. Okay, so they did it in a short time because of the synthetic biology tools. Okay, so these are some of the information about a lot of effort been made. More than 135 coronavirus vaccine projects are currently underway because the coronavirus has so many variants. So you can alter the code to fit for the right vaccine for right product. Okay. So just to give you an example, the conventional time frame for making vaccine, varicella 20-80 years, flu missed vaccine 20-80 years, human papillomavirus 15 years, rotavirus 15 years, COVID vaccine 12 months. Okay, so this is one of the advantages of synthetic biology. You shorten the time frame to make the vaccine in such a short time. So to make a long story short, when you make the vaccine, we are looking for this spike, viral spike, and that spike is what attached to your lung cells caused infection. If the virus cannot attach, it cannot cause infection. So you are making code to make that code is a wrong code in the virus, so you won't attach it. That's what that viral vaccine is. It's a long story about COVID-19 that we don't want to relive that. I'm just going to skip all this part. This is how the vaccine was developed. Okay. So we'll skip to the virus here. Okay. So that's how a vaccine look like. We are concentrating on the spikes and the virus. So we look at a recombinant vaccine. You take the code for that spike and you put that in the messenger RNA expression. When you take that, your body make antibodies to this virus and it won't attach you to yourself and you avoid infection. Okay, that's what the messenger RNA recombinant vaccine does. So the story of all the vaccine production, different viruses are here, too much information, so you can read on your own time. It will be available for you. As a part of synthetic biology, messenger RNA vaccine is what the two companies, Moderna and BioNTech and Pfizer, they're all working on, and they're constantly tinkering with it for new variant. Okay, it takes that several trials to make your vaccine approved, phase 1, phase 2, phase 3. Okay, this was achieved amazingly in 1 year, and also the urgency of it, because a lot of people are dying at the time, so the government approved it faster too. Okay. So the last part is regulation. So we are making this all kinds of stuff, so it has to be regulated in a right way, so it won't end up in wrong hands, and it could be a serious problem. Like poliovirus, people can create poliovirus. Okay, the risk of synthetic biology posed to human health and environment are serious, since synthetic biology has the ability to create organisms that have never existed before, and the complexity will only increase over time. So we must establish proper regulation safeguard before this technology evolves too far and will be too late to control it. Okay, I think that's the last slide. I'll stop here. So that's in a nutshell for you what is synthetic biology. In one sentence, you're creating new thing by putting the code that you already know. The beauty of that is the codes are synthetically made. It's not from nature. You make those adenine, thymine, cytosine, guanine. You make it in your test tube. That's what it is. Okay. Let me stop here and take questions if you have. Yeah. Yeah.
And there are a lot of vaccines to just straightforward this problem.
Yeah.
And but is there any method developed to just stop this unintentional escape of viruses and synthetically developed organisms from the lab itself?
Yeah, all these places are very controlled lab. Is very safety, high level safety. Very rare that it could escape out. Okay, but I mean, accident can happen. Oversight. Sometimes people make mistake. That's where the worry is. Okay, and then there is another problem in the U.S. They always worry about the intellectual property theft, and people come and work for you and steal, and go back to the country and create problem. And that question is lingering in the U.S. They're worried about that intellectual property theft. People come looking for job. They are smart people, but their thinking is totally different. They want to know the technique, learn the process, go back and make it, and it could end up in a wrong hand. So yeah, but they try to make it safe as possible. You cannot go into those labs. It's really tight. Okay, yeah.
But scientists with goodwill will develop these viruses in great safety and all.
Yeah.
But there may be people who are just focused on just destroying the human culture. There may be people like that.
Yeah.
So how to just change this thought of
Yeah, I mean, if you want to make this, you need to have right skill. The guardian angel here is the people that want to destroy. Hopefully, they don't have the skill to make it. These are all, you and I cannot make it. You got to have years and years of training to make this. Okay, that's my one safeguard. Right? But on the other hand, people with intention, smart, but they have a bad intention. They go and learn and they can create it. That's the one we worry about. Yeah. Okay. Any other questions?
Sir,
Yeah.
sir, good evening. I have one question, sir. Sir, related to genetic engineering
Yes.
or synthetic biology, algae has been modified outside India for extracellular secretion of lipid. Is there any literature, work?
I didn't come across an algae work about the lipid part. There may be people have done something, but I didn't come across. Maybe you can do a literature search. There may be. All you need to put the keyword algae, lipid, synthetic biology. Google will pop
Yes,
up.
yes.
Yeah.
Yes.
I don't know. I never come across anything myself. Yeah.
Okay.
Yeah.
Thank you, sir.
Maybe you can create one. Okay. This is the time to do it. Yeah.
Yes, sir. But it is the need of the day
Yeah.
to secrete the lipid outside.
Yeah, you take the right training
Extracellular.
technique
Yes.
and training, learn the technique, you can create your own tailor-made
Yes.
cells. Yeah. Yeah.
Thank you, sir.
Oh, you're welcome. Yeah, you have a question?
Thank you for your wonderful insights on synthetic biology, sir. My question was on this precision medicine.
Yeah.
The traditional medicines are not found that effective or that effective on treating the diseases. So there was a paradigm shift towards mRNA vaccines. So do you see any future of this synthetic biology in this mRNA-based vaccination, like we did in the COVID case?
Yeah.
So is there any scope of synthetic biology towards mRNA-based vaccinations, or
Is there a scope for people to make similar mRNA type of vaccine? Is that your question?
yes, sir? The challenge, particularly to our precision medicine,
Yeah.
the tradition or allopathic medicines that lack targeting the sites, disease-causing sites.
Yeah. Yeah.
So mRNA vaccines have found more suitable or more effective in
Yeah,
those things.
yeah.
So
You can use this.
one of the application of this synthetic biology can be
Find the right place to make this adjustment, yeah?
Yes, sir.
Definitely, yeah. Yeah, synthetic, that's the beauty of it. So if you know the code, if you know the function, if you know exactly the target, you can create this cocktail together and put together, yeah? It's possible. Synthetic biology can make it very precision medicine that you're talking about.
Yes, yes.
Yeah, yeah.
Thank you, sir.
Yeah. I mean, in this bioenergy conference, I mean, I gave an overview of synthetic biology, but you can tailor make your cell to produce better yield for, you know, your biodiesel, ethanol, any product you want, you can make it. Like I told you, rubber, you know, precursor rubber molecule they're making, isoprene, right? Anything you want. So you go learn about it, about synthetic biology, and train yourself. You know, you could be a billionaire. Okay, all right. Thank you for your time.